When you think of the brain as a computer, you usually think of neurons as individual decision-making nodes. They receive a lot of different inputs and decide to fire or not at each moment. But zooming in, we find that individual synapses, the chemical junctions by which one neuron signals another, are complex analog computers in their own right. They receive all kinds of chemical, electrical, and biological information and decide whether to let a pair of neurons talk to each other. Together, synapses shape the flow of information across the brain. When the system goes awry, neurological and cognitive symptoms can manifest throughout the entire brain in conditions such as autism, schizophrenia, ADHD, even possibly depression. Perturbing it with a chemical, on the other hand, can bring about extensive cognitive change, be it psychiatric treatments or psychedelic drugs. How do synapses do it? Each synapse is built from a set of hundreds of thousands of different proteins, little molecular workers, each with their own crucial job. All these workers depend on each other in complex ways. Pull on one thread, and a dozen others will change in seemingly unpredictable but actually well defined ways. As a whole, this network of interactions and dependencies shapes the rules by which synapses function. Figuring out who's who may feel like trying to understand all the relationship in a soap opera. Who are enemies, who are friends, who's the real father. And in the soap, like in biology, you need to actually watch it play out. In my research, I work on mapping out this network by looking at many proteins simultaneously in single synapses, relying on a nifty trick involving DNA barcoded antibodies that connect to each protein. To figure out how the different proteins measured depend on each other, I adapted a mathematical tool called Bayesian network inference to draw a map of who depends on whom. This model gives accurate predictions. Turn one knob, and you can know how the others will turn. I then turned to a long-standing question in the genetics of autism and schizophrenia. A lot of different mutations can lead to autism, but how can mutations in so many different and unrelated genes lead to the same symptoms? I looked at how this network is affected when you mess with different genes and saw that some parts of the network are affected the same way no matter which gene you change. This can be a clue to what happens in these disorders at a molecular level. I'm now working on measuring how this network is affected by different psychiatric drugs, which may help humanity take on the notoriously frustrating challenge of rational, informed psychiatric drug design. In the longer term, I hope these kinds of insights will contribute to a deeper understanding of the brain as a chemical and not just electric computer.